through genetic manipulation of root system agriculture. Dr. Menamo research utilizes a combination of advanced molecular techniques, bioinformatics analysis, and field-based experiments to identify and characterize key genes and genetic location controlling RSA in cereal crops, particularly sorghum, wheat, and barley. He published several peer-reviewed articles in reputable journals. Dr. Minamo's research aimed to contribute to the development of cereal varieties with improved root system for enhanced nutrient and water intake and resilience against abiotic stress. Dr. Minamo's presentation today entailed multi-locus genome-wide associated for identify group adaptive alleles. He had 30 minutes for his presentation and another 15 minutes for answer our question. Thank you very much, Dr. Temesgen. You can start. You can start now.
Okay, thank you, Professor Zad. I would also like to thank you, the organizer, to prepare these seminars. I will start my presentation seminars on multi-locus genome-wide association study for identifying crop adaptive allele. So my name is Temesgen. Also, Professor Elzaid already introduced myself. I will directly go to the outline. So in this seminar, maybe 30-minute seminar, I will give you some highlight. I know most of the participants, they know about what's GWAS something, so I will not go to in more detail, but I will highlight first in introduction. And I will also give you some highlights between this multi-locus and single locus, and I will give you some why single locus has some limitation to move to this multi-locus GWAS method. Then I will give you some method for doing a multi-locus GWAS, and I will focus on the importance of adaptive allele in the crop improvement, particularly in cereals and some case studies. And I will also give you how can we do a multi-locus for finding of adaptive allele in specific crops. And I will give you some case study as example on sorghum, charo, and lima bean to identify the adaptive allele using this multi-locus. And finally, I will give you some future perspective and conclusion parts. So I think majority of the audience are from genetics part. They know what is GWAS. GWAS is a population level genomic approach that scan almost near to 1,000 to millions of genetic markers, particularly using these SNPs across the whole genome of a specific crop to identify association with a phenotypic trait. Like example, we can focus on disease resistance, droughts, and climate adaptations. So when do GWAS actually, we have two options to analysis genomic wide association study or analysis. One is just using a single locus method, and the other one we focus a multi-locus GWAS. So when we look this a single method, let me. Yeah. So when you use a single locus, actually doing associations between each single the nucleotide or SNPs variations a trait at one time. Most of the time when you do a single locus, we are using two models as general linear model and mixed linear model. So when you use this one, we can use actually in the different software like R, and also we can use most common one is a tassel softwares for this one. But when you use this a single locus, actually very strange identifying significant threshold or using a multi-test correlations. So there are three different methods to define our threshold. The first one is we call it a Bonifer correlation. That's a very strange type. That the formula is like dividing the p-value, that is either 0.01 or 0.05 divided by the total markers just finished. So this type of the threshold is very strange and really very strict. So most of the time it will be very difficult to identify specifically allele for climate adaptation for adaptive allele, because later we will see the adaptive allele, they are controlled by many genes, they have very small contribution, so such type of threshold may not be identified. Even the second one is threshold using CDAC correction. This one is less stringent than bonifer corrections. That's the formula is like 1 minus just as minus 1 minus alpha. That's either 0.05 and the square root of 1 minus marker, number of markers, same as a marker. So here also still use the total number of marker, and it's when you do, it's less stringent than bonifer, but still using this type of threshold still have a stringent. And the last one is for a single marker analysis, L and J. This one is less stringent, but why it use rather than the total marker, it use only the effective marker using the for threshold. So to define this effective marker, this L and G define some package. You can access it from online. That's, we call it in R, MAF package. So you can get this one and define the effective marker. So on the threshold definition, they used, rather than the total marker, they used this effective marker. So for example, when you see, let's assume if you have a total of 300 marker, you can look the difference, the threshold. In bonifer, it's very high, stranger, but when you compare to CDAR, it's a little bit better, more, it's higher p-value. And when you go to this one also, it's better. So when you go to bonifer 2, this is the last one. The stranger is more, bonifer is very stranger one to define. So using this single marker to identify these adaptive alleles, particularly adaptive for specific climates, very, very difficult using this threshold. So this is the example what make better other than that. Bonifer is very conservative type. CDAR is less stranger, is less stranger and less conservative when you compare to bonifer. And LG is less stranger and less conservative compared to the either. So at least the LNG, but for adaptive allele is not such important. So what's the limitation? So when you do particularly for adaptive allele in single locus GWAS, the first one is high false negative in a single locus QTL. Like example, if you define using the threshold bonifer method, it's very stringent. We cannot detect alleles which has very small effect in QTL, particularly in adaptive allele. Most of the alleles have very small effects. The other one is ignore epistatic. So the interaction between different gene or different alleles. Population structure biasness and low power for polygenic trait. So it has weak detection, the trait which has controlled by many genes with minor allele effect. So for this, we will move to this multi-locus to detect particularly for this many alleles. So what is this multi-locus GWAS actually? So this multi-locus is define statistical power, simultaneously analyze multi-genetic loci to identify association with complex traits like adaptive allele. So when you look this graph, so there will be a lot of, when you look based on the single allele loci GWAS, so it looks like almost all what you're observing this marker will be significant, but the multi-locus will identify only the alleles which has, later we'll discuss in detail, identified by multi-genome as well as have a multi-effect for that a given trait. So what is the importance to identify this adaptive allele in crop? So now the crop actually, when you look, they have a survival. They can survival environment. So we have to, when you identify this adaptive allele, it can help us to identify a survival for different stress. Like we can identify biotic stress like drought, salinity, heat, biotic stress, as well as the changing of climate conditions. So because we are later we'll look the individual which we use to analyze the multi-locus, so they have ability to survive that environment. So we have to identify which allele make those individuals to survive that changing environment. For this hour today, we'll focus on to analyze multi-locus for changing climate conditions. So you can look, sometimes you look at some type of crop or plants which survive and which the environment is not suitable. So in adaptive allele, it makes, we'll look what makes these individuals make survival. So we'll identify that alleles. So to do this multi-locus, actually, there are a lot of popular models to do a multi-locus GWAS. It's most of common are this is PharmaCPU, Blink, MLML, and Lasso and ElasticNet, and also others. So we'll see some of them, those models. So why we are looking rather than the single locus to multi-locus GWAS for adaptive traits? As I mentioned, one of the big challenge in adaptive traits is that a polygenic adaptation. The adaptation is not from a single gene. It's from an effect of many genes with minor effects. So we have to identify those alleles. So single locus, that may be a challenge to identify using single locus due to very strange type of threshold. The other one is the behavior of this epistatis, gene-to-gene interaction. So that's also is very difficult to identify using a single locus. So we have to move to this multi-locus. And reduce false negative, because if you remember in the previous slide, I said that single locus has very strange, so there are a lot of false negative results. So that will be solved when you use a negative, when you use a multi-locus GWAS. And the other one is when you use a multi-locus, we can integrate the environmental data as well, rather than using only the genotype data. We can also incorporate the environmental variables like temperature, precipitation, and other to detect a climate association selection signals, and also to detect which variables or which climate variable contribute to for the genetic variation of the individual. So this is the examples we did on the sorghums using some agroecology data, like the collection is collected from different parts of Ethiopia with different agroecology. So we used agroecology as a factor, and we also used the geographical locations, a distance from one into other, used as a variable, and how these two, agroecology and geographical location, contribute to the variation for those genotypes or SNPs data. So you can will observe like this. For instance, when you look, agroecologists contributed almost 7%. When you look the interaction, almost 3%, and locations, geographical location, it's contributed almost 3%. So we can look using a multi-GWAS to detect also this type of. So how can we do? That's the big presentations for today to do the GWAS. First, we have to get a phenotype data or climatic data. How can we get this one? We'll see some examples to get this climatic data. The second one, as a GWAS, we need genotypes. So this genotype should be 1,000 to 1,000,000 base SNPs markers, because we need high density marker to detect particularly very small allele effect. Then we should also do a population structures using a PCA or kinship matrix we can do. And the multi-locus model will example look as this MLA model, and this for today. And finally, we'll do a candidate gene identification. So to do identification, first we have to do LDDK, then we'll see how to use multi-locus GWAS to identify a candidate gene for that specific adaptive alleles. So the first, as I said, how can we get a phenotype data, the first one, particularly for climatic variables? The source should be a local adaptive land races. We have to collect land races for that a given location, but this land races should have a GPS or the geographical location information, very important. So based on that geography locations and different collections, then we can extract a climatical data. This is one example. You can get a climatical data from all lines, from different satellite source, from different GPS source. This is the example we can get from world climate data. So this example, when you take this source, it has two different type of climatic information. The first one, historical climate data. When you go to this website, so you'll get the climatic data, that's temperature, rainfall, and other the climatic data from 1970 to 200, 2000 to 2000, 2021 to 2014. So you'll get, this is as a current, this is as historical data. So we can use this as historical data, and we can look which alleles are more related for that specific climate data. And also there is also a future data, some information you can get what will be the future. So there have a predicted environmental data like from 2041 to 2060 and from 2061 to 20. They have the average of this data in climatic data, so you can use this climatic data to find adaptive area. So this climatic data, as I said, that there may be temperature or precipitation and even solar radiation, wind speed, and other, even water vapor pressure and other data. And there is also, you can use in different geographical spatial resolutions, like from 10 minutes to 30 seconds. 30 seconds, almost within 1 kilometer square, and the maximum 10 minutes, that's 340 kilometers square. But this is very small, this is very large. Then most of the time we recommend to use around 2.5 minutes is very, very good. The rest will be very wide and the other one is very, very small. So when you collect the genetic data, we have to consider these resolutions. And other climate data, when you click these bioclimatic variables, you'll get almost 19 different type of climate data related to temperature and precipitations. So look, this one from bio 01 to bio 11 is related to temperature, like annual temperature, mean diurnal range, isothermal, temperature season, maximum temperature for warmest months, so minimum temperature for coldest months. It is continue like this. This is in the temperature in different format. From starting 13 here, you'll get here from 13, you'll get a precipitations, annual precipitations, precipitation wet estimates, precipitation dry wet. So almost all are precipitation, but indifferent. So this one helps us start which type of temperature or which type of precipitation can help to survive that a given, and so we can identify allele for this a given variables. How can we get those? Actually, that's online available. So we can extract using some R software or R package, which is called, this package, geodata. So you can use this library, then you can extract those data. How can we extract? Using this function. For example, if you use this, the third functions, world climate titles. So we can use a variable. This variable has either our temperature, precipitation, or that 19 bioclimate data. Then we can use the GPS location here, longitude and latitude. Then using the longitude and latitude, we can extract this data directly from online available sources. So different type of, as I mentioned, we can extract this bioclimatic 19 bioclimatic data, or we can extract from online this like temperature and precipitations data. So this is the data how to access. Once we get this access, first we have to look which variables contribute for a variation for our genomic data. So we will do a redundant analysis. So this redundant analysis, we can use a function of this library of, if you look here, we can use vegan library. Then we can calculate, or we can see by changing of that variables, we can look which one has a contribution for variation of a given genomic data. So that we do that based on this function, or we can also use another way, what I told you in the previous presentation. We can put some variable here, we can put another variable here, even we can make more variable. And which one is contribution? How many percentage can contribute to this, each variable for changing for that genomic data? So they're very important. Once we get some information, then we'll pass to multi-locus GWAS analysis. So to do a multi-locus, so as I said that we need this genotype data. This genotype data should be thousands of millions of SNPs because it needs high density marker to do a multi-locus for adaptive allele. Then once we have that markers, then we have to also look at population structure. This is example one of the software we will do a multi-locus. So this software, it uses a population structure in different format. Here, one is we can use a matrix, a population matrix, or we can use a PCA population, or we can use evolutionary population structure. So we have to do this one in Tassel or in R. Then we have to produce, this is a PCA for instance, and this is a Q matrix. Then we have to use this one as a population structure to control the population structures. Once we have this one, we'll do directly a multi-locus different model. So there are a lot of packages in R to do this multi-locus. So we will focusing on this one. Most of the time we are using this MR-MLM model is very, very nice and other, but we can use also another this package. So this is one of the package, RMFV. So when you look here, the method use different method like JLM, MLM is one a single, but for this model, we'll use a pharma CPU as a multi-model. The other one, GAPIT, you can look, there are at least here 3 different type of multi-model analysis. You can select this one to do our analysis. The other one is the most recently we are using one is this MLM model. This is a multi-model. This one had almost 6 different type of multi-locus GWAS. So this software, it did using 6 this model, and the threshold is at a load score of 3. That is almost a p-value, almost less than 0.0. At a threshold is 0.001. Is put. We can make it more, a little more, but this is as a minimum threshold for a multi-locus. So after doing this, you'll have an output. Look, detected a marker detected by different model. Here we cannot consider all marker which detected by a single. Look here, when you use this model, one marker it's detected by different models. So for instance, this marker is detected almost using 5 models. But look, this one only detected 1 model here. So for further analysis, now rather than focusing on the individual marker which is detected by model, we'll focus on the markers which detected by very many models like this. So this is one of the advantage of using this MLA model compared to other models. So then finally we'll go to the candidate genes, because once we identify those alleles, just we cannot stop on alleles, so we have to go to further which genes are associated with these markers. So to define this one, first we have to do LED decay. So to do LED decay, we should analyze the linkage disequilibrium. We can do this one using tassel as well as we can do R. So once we have this one, we'll use in R package. Using this R package, we'll define what is the point LED decay happening in different populations. This is in different populations which have experience in different crop. So this LED decay gives us the threshold or the starting, the up and the downstream of the alleles, which we focus on the genes. So based on this gene, we will go to identification of genes on the website, which is called a phytosome website. We'll go to, for example, there is a lot of the bioinformatic data, but I'm just focusing on the phytosome is very easy. So you can go to phytosome and particularly this biomart, and you will define your crop, and you have the starting and the end point of the genomic region. You know which chromosome is on that specific crop, then put it here, the position, because based on the LED, we can put it the starting end position. We know the chromosome region. Then we are make search, then we'll know the number of genes around that LED for that specific significant markers. So this is how to do, but I will give you some example as a case study we did in different crop like sorghum, taro, and lima bean. In sorghum, we did almost 1,000 of collection from Ethiopia in different. This is the agroecology of Ethiopia. We collected. This is our collection. We collected sorghum from almost Ethiopian producing regions. This is the production area of Ethiopia. Then we collected based on this production area. We collected sorghum different region, and we also all this collection have geographical information. Based on that geographical information, we extracted from online the temperature and the precipitations annual. Just we focus the annual temperature and annual precipitation, and we also use altitude as a factor to do a GWAS. Then we did a GWAS and identified very alleles which are contributed for these three different climatic data. And we try to also look any collocation from previous data. So we obtained this one, have some of alleles are linked to previously identified cold adaptation key. So that's what we are interested to look adaptation for specific cold, specific for drought. So we are looking that we get these adaptations, the alleles which is collocated for adaptation called adaptation, and some of them are for drought adaptations. Similarly, we also did on the taro for Nigerian collections, and the collection in different regions of Nigeria. This is the collection regions we collected are different regions, and we also identified, we did use a different biological, biochemical, bioclimate data like Bio1, Bio2, this is for temperature and precipitation. And we also identified a lot of markers which is associated for that climatic data. Then we looked, we did LED decay specific for that crop, and we find some genes which is associated with this data like for temperature as well as for precipitation, and we'll align with what is the function of that genes. Most of the genes are important for this estrogenic, dimethane dicarbonate, and as myosin transcription factor, which has more association for climatic adaptation also. This is one of indications to alleles for that adaptive value. And the last one is lima bean. We are this not published one. Currently, we are working on that once. This lima bean is collected from Ethiopia production area. Not only Ethiopia, actually, we collected from different countries. Collected this. All this one, they have geographical referred information. Then based on that, we extracted all climate data as related to temperature as well as for precipitation. So this data classified into two. There is historical data from the average of in 1970 to 2000, and the current from 2000 to 2040, and the last one future 2024 and 2016. So what was the result? Then we did a multi-locus GWAS, and we identified several loci which are associated with in different historical, from historical and from current and for futures data. Then we just look at there any details which is associated or stable across these different time period, historical curve. This is the whole markers. So we identify almost 83 of them are found in all climate data, in historical, as well as in current, as well as in the future. And we also selected only the marker which has identified in different models from 6, at least 3 models detected. From that ones, out of this, almost only 22 markers are more stable. That means they are adaptable in different in different historical, current, and the future climate variables. So this indicates that this marker may have something for adapted of this limb bin. So this is actually not published, so we'll publish soon. And we do also LED decay. Almost we obtain 150 kb as LED decay. Then we find several genes around this one. And among that, almost 12, near to 12 genes are more associated with the markers which found more associated for climate adapted genes here. This is some loci which is more associated with adaptive alleles. So this is what I have. In the future, this is as a recommendation, like integration, like a new technology like machine learning, considering pangenomic to identify very rare allele. High throughput phenotype is very important to use a drone. And for precision agriculture, so we have to custom allele stacking for climate resilience to consider this adapted. So in conclusion, so when you do a multi-locus allele, it's better for adaptive allele because of single allele, very stringent to do a GWAS. So multi-locus is better to identify this adaptive allele, particularly because adaptive allele is controlled by minor allele effect. So minor allele effect may not be detected through using a single locus GWAS. So this is enable the fasting crop breeding for climate resilience. The future work combining multi-locus for genomic editing is very important, and identifying for integration of this trait. So this all about, I have welcomed the questions and answers. Thank you.
Thank you for following us. We sincerely thank you for your continued support and engagement across all our platforms. Your trust means everything. Stay connected. Together, we grow. Thank you for following us. We sincerely thank you for your continued support and engagement across all our platforms. Your trust means everything. Stay connected. Together, we grow.